Welcome back to the Medical Innovation Podcast. My name is Pranav, here with my co-host Arvind. Today we're really excited to be speaking to Mathai. Could you please introduce yourself and tell us a little bit about your background?
Sure thing. I'm currently the CEO and chairman of Parabilis Medicines. I've been there about 2.5 years. Prior to that, I ran the global R&D organization at Johnson and Johnson. I was there for about almost 6 years, and I've had a career in business and research and development. So prior to J&J, I ran most of the research at Merck. And prior to that, I had started a company called Theravance in the Bay Area, and that was based on my thesis work at Harvard University prior, and that was all those experiences were immense experiences, huge learnings, ability to make medicines, ability to impact lives.
Yeah, it's incredible. And I think we're reading a lot about what you're doing at Parabilis right now. And I think it's a really unique take, especially drawing from all your experiences. Would love to hear how you kind of transition from the other drug development, companies that you've worked with and taking your experience that ultimately led you to work with Parabilis.
Yep. So as I said, I've been very devoted to elevating health for all people, both in the United States, elsewhere throughout the globe, and always aim to make medicines that are really effective, like high effect size. Sometimes in clinical trials they say, and so being really choosy over the years on what to work on. And, through my career prior to Parabilis, I had the privilege of bringing forward 18 different medicines. Many of them are practice changing. And so the bar is high for me of what's worthwhile working on. What brought me to Parabilis was the technology that you're referring to. And it is a helical peptide technology. We call it a Helicon platform. And what it does is it opens a door to certain kinds of proteins that are inside cells that are otherwise completely inaccessible. And that's a big deal because it turns out that inaccessible or undruggable proteins constitute about 80% of
Wow.
biology. So it's a huge swath of all biology. And much of this then gets connected through genetics or phenotypic screens, um, a piece of biology and a cause of a disease. Much of that then is unprogressible. It's terrible. These patients that have that particular disease connected to an undruggable target are left indefinitely to suffer and often die. And so that's a big deal. It's not really connected to rare or common diseases. Actually, it's 80% of medicine, I would say,
Yeah.
across all therapeutic areas. So it's the majority of what you can imagine as illness. And so what Parabilis did is based on the idea of Greg Verdine, Professor Greg Verdine at Harvard. He's been working on a concept of alpha helical or stapled peptides, constrained peptides in a particular shape, this corkscrew shaped structure. He's been thinking about that for probably 25 years. And about 10 years ago, all that crystallized for Greg into a company that he called Fog Pharma, now called Parabilis. So, same company. And that idea was based on the following notion that inside cells, uh, only small molecules can be used right now to access targets inside cells. You can't have antibodies because antibodies don't cross the cell membrane. Now, for small molecules, they all require a small pocket on proteins in general to have any activity. And most of those, the 80% that I'm referring to, don't have pockets. And so that's a real problem. And what do you do? So what we've done, what Greg envisioned and what we've put into practice now is creating these alpha helical peptides that function almost like antibodies. So they can bind relatively flat or low curvature surfaces and access all those proteins that otherwise you couldn't get to with a small molecule. So that's the technology. How to actually do that is much harder than it sounds.
Yeah.
Uh, you know, it, in the old days, that was an idea that was tried to people tried to prosecute it by using natural peptides, and and they would often use a staple, but it turns out there's not enough diversity in a natural peptide to be able to gently go through membranes.
Yeah.
Instead, it was necessary in the end to put fairly harsh cell penetrating mechanisms like lots of positive charge or big fat tails, and these rip through the membrane, but those are never going to be drugs.
Yeah.
So what we did at Parabilis post it starting is start to introduce into the peptide the full diversity, the full power of small molecule discovery by using unnatural amino acids. So change it from the 20 natural amino acids to thousands. And when you do that, you introduce a massive diversity, like an astronomical diversity to these peptides, so that you can actually co optimize for passive, gentle entry into a cell without any harm, and yet have the diversity to co-optimize for binding. And that trick, of using unnatural amino acids on a peptide backbone turned out to be the core of the idea. We've since needed to reduce it to practice in additional ways. We use a significant number of computational algorithms, both physics-based and machine learning-based. So the AI revolution has been incredible for Parabilis, because we've generated a ton of training data inside the company that we use to train algorithms that then help us predict cell entry and binding and solubility and all the other features you need in a proper medicine. So that way we've opened the doors. And so now the doors open, we're doing things with it that are pretty phenomenal, tackling targets that have long been considered to be absolutely off the table, undruggable. And the first of these is beta catenin, and we can talk more about that. But each of our projects in the company is special. We're the pioneer. There's no replicate of any of our projects anywhere in the biopharmaceutical industry, which is unique. Like there aren't any companies like that I know of, that the entire portfolio is unique. Not only is it unique, but even if one tried to follow it, it's very difficult because these targets are not typically drugged. And so we have access, obviously, through tremendous amount of know-how, how to design based on what I mentioned about the technology, but others don't. So between know-how and intellectual property in patents, we're uniquely situated to prosecute this platform and tackle the targets and tackle, therefore, the diseases that we do. So it's a beautiful setup, and the company's really humming at the moment. The platform's working well, and the first of these projects is yielding clinical data that's very compelling.
Yeah.
And there are two more projects that are coming into the clinic in the coming period.
That's amazing to hear. Like, you're essentially creating a new way to impact the way the body functions. Like, the majority of medications today, they can't go with the inside the cell barrier. They just act on the cell and try to trigger some action within the cell. So, we're actually being able to go inside the cell and manipulate how the proteins are functioning. That's a very big impact, and there's a lot of
It's huge.
potential there.
Yeah.
It's huge.
You mentioned beta catenin and a couple of other products that you're starting off with. I'm curious, how did you prioritize? What were your
Yeah.
criteria in deciding what to prioritize first?
Yeah. So the number one philosophy that I would have is pick mechanisms that where there's confidence that they're going to make a huge difference to a particular disease. So don't bother with anything incremental. Um, so there are lots of when a company does something new, when there's an AI and drug discovery approach or a potential new modality, the 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 draw is to do something that's extremely well known and validated, so that the only risk you take is in the technology or the
Right.
approach itself.
Right.
But you end up always in situations where you've made something rather incremental, and we'd rather not do that.
Yeah.
And so all of our projects are big swings. And I don't mean to make it sound risky, because there's so much validation around the genetics or phenotypically through other mechanisms of confidence, so much confidence around these targets, it shouldn't be seen as some big risk. But yet it's connected to a high effect size medicine, a place where we can really make a big difference to patients, as opposed to just offering something, slightly incremental. So that was the number one criterion, big effect size.
Right.
Second, it shouldn't be a protein that in principle even is addressable by any other modality. Because if you could, go do that
Yeah.
in a simpler way. So it should be one of those 80%, which is uniquely addressable by Parabilis. And third, it should be possible that we're meeting an unmet need right now that's profoundly you know needy, so that the patients have a dire circumstance that they're in. They're in exquisite pain or discomfort. They're at risk of death. There's some very significant suffering associated with those medicines.
Yeah.
That makes sense to me. Like, that's the place I'd rather approach first. There are other kinds of medicines you could make. Of course, like we're following the obesity medicines right now, but those kinds of medicines are not my first port of call. They're going to be medicines where you're in dire need.
Right.
And so high effect size, dire need, and uniquely progressible
Yeah.
by Parabilis' technology.
I think what's really exciting about this is the concept that you're spinning a company off of a platform. And I really want to talk about fog and the data you talked about at ESPO. But before that, I can understand your excitement around this because I'm assuming there's many of these pathways you've already thought about in the drug development side of like these are diseases that we can know that we target. We're starting
Yeah,
with fog 001, but we have all these in the pipeline because we have this novel new
no
platform to
question.
then implement it. So I can see the excitement,
Oh, yeah.
behind that.
Actually, it's very difficult because it's very difficult to contain the
You can't
energy in
tell
a
us.
sense, because we have so many ideas that
Yeah.
we'd like to work on, but it's very important for our young company to stay disciplined and not take on like 20
Yeah.
projects.
Exactly.
So we've taken on several projects, each of which can make a profound difference to patients. And we need to prove ourselves through those before we expand the
Right.
aperture and take on additional
Right.
projects.
So if we start with fog 001, this is a medication that you're planning on using to target desmoid tumors in particular. Would love to hear first, what is a desmoid tumor? What are these patients that are affected by this type of tumor? How are they presenting? And then ultimately how you're planning on addressing this
Yep.
with fog.
So first of all, this target,
Mhm.
beta-catenin, it's part of the Wnt beta-catenin pathway. There's a single node in that pathway that people have known for many years is the critical node, and that's the interaction between beta-catenin and the TCFs. They're the T cell factors that are transcription factors. That's the node. Everyone knows it's the node.
Yeah.
However, I think every company, including my own team at J&J, my own team when I was at Merck, tried really hard to throw the book at this through
Yeah.
collaborations. The corporate deck screened in every which way you can imagine, and never yielded anything actionable. So it was always ended in disappointment. So that node is what we went after. And the node's been known since Bert Vogelstein discovered it in 1990.
Wow.
So it's been associated with cancer for a very long time. So that it's been drugged at all with a highly selective, very potent inhibitor is a massive achievement.
Yeah.
It's truly drugging the undruggable. Now that, then we have choices of where to go. The way we conducted our trial, and this is public information, is we looked at any patient that had a mutation in the Wnt beta-catenin pathway, where that mutation ended up turning it on in its pathological way, like huge flux through the Wnt pathway. And so there are numerous cancers like that. There's probably millions of patients a year that have beta-catenin associated tumors. We picked some, and some of them we've talked about, some of them we have not yet. Desmoid is the data set that we released at ESMO in Berlin just last week. And desmoid tumors are driven by beta-catenin predominantly. They sometimes can be driven also by an APC mutation, which is the flip side of beta-catenin in the pathway, and both equally addressable by our medicine. And desmoid tumors, they can show up in any part of your body, and it's quite horrible when you have them because they just grow relentlessly. And the abdominal ones are associated with exquisite pain, and it's almost this intractable pain that there's no pain medications that can really control that level of pain. We had one patient where the desmoid tumor was growing in the person's throat, so it was deviating the trachea, and they were having more and more difficulty breathing. There was another patient where it was wrapped around the jejunum, the small bowel, and was giving intermittent small bowel obstructions. There was another patient where it was in the leg, and the person basically lost use of that leg and had horrible pain. So each of these have their own story depending on where the tumor shows up, and it's pretty awful, and it's chronically with you, and it just grows. So what we showed at ESMO is that we treated a number of patients. We had virtually uniform response in all the patients, and we had a 83% what's called an ORR rate, which is the objective response rate for patients after 4 months of treatment. That's phenomenal. So 100% disease control rate and an 83% ORR rate.
Wow.
So that is what I was saying a few minutes ago when talking about high effect size, uniform response, everyone gets a lot better. That's what we're aiming to do for these patients with desmoid tumors. We had a company offsite a couple weeks ago where a woman with a desmoid tumor, this is the one I mentioned just a minute ago, that had a tumor wrapped around the jejunum.
Yeah.
And the way she describes is it's just awful. It's been with her for so many years. And you can tell, like listening to her, that she's really beaten down. And that kind of person, that kind of patient, we can help that person and we can make their life a lot better. She's married and has children, and the entire family is affected by her illness. And everyone kind of is in this swirl. We can help all of them. And so it's the patient and everyone around the patient. So we're very, very happy about that. There are going to be additional diseases that are beta catenin or APC driven, and we'll be talking about data there in the coming period,
Yeah.
months, and over the coming years, even as we expand what we're doing with beta catenin inhibitor.
And with this medication in particular, we were talking about this earlier about how the Helicon technology you're using is a very targeted therapy. You're able to get through cells in a non destructive way, target the exact protein. So in theory, it sounds like this is a safe medication.
We also discussed the incredibly good safety and tolerability data associated with the medicine at doses that are very effective
Yeah.
at the ESMO meeting in Berlin. So that's part and parcel why we're so excited.
Yeah.
Not only is it uniformly active and highly effective, but it's a well managed, well tolerated medicine. We're going to administer it once it gets to the marketplace in a sub-Q auto-injector,
Oh,
and that's much like the GLP-1s are administered
Yeah.
today. It's very convenient, every week or two. So it's going to be a life-changing medicine for the group of patients that we're talking about here.
It's awesome.
that's fascinating. I think desmoid tumors definitely meet all three of your criteria: the high prevalence, high impact, and it's something that no one else can treat as of right now without using your patented unique technology. So I'm curious, what does the long term look like for Parabilis? You mentioned you're doing a couple of disease states initially. You're going to expand beyond that. You'll do more beta catenin tumors down the road as well, or conditions down the road.
Yep.
Do you ever foresee, for example, you have a brand new technology, so would you ever consider licensing it or trying to broaden your impact in that way?
Great questions. So, first of all, with the beta catenin, one, the beta catenin inhibitor, yes, the idea is to continue expanding its use beyond desmoid tumors to other tumors that are driven by beta catenin and APC. So that will happen. And these are both rare tumors and very common tumors.
Right.
And so all of that will be covered, and we'll build it. We'll have multiple indications for the same medication in the label. So there's two projects right behind it that both can target the prostate space. And
Very
prostate
much needed.
cancer is an area I've spent a lot of time on at Johnson and Johnson. We were heavily into prostate cancer, and so it's an area I know very, very well, and what the unmet need might look like. There's a target called ERG. It shows up in about 40 to 50% of men with metastatic CRPC, this castration-resistant prostate cancer. And those men, if they have that ERG-TMPRSS2 fusion, they do much worse. If you look at Kaplan-Meier plots, they separate very quickly. So there's a much worse outcome if you carry that fusion mutation. So people have hypothesized that ERG is the driver, and is a great target for prostate cancer for many years. But it's almost this round, orange-shaped protein. There's
Yeah.
nowhere to grab it, and it's a classic undruggable protein. It's one of those large portion inside cells. Um, Professor Charles Sawyers in New York has been working on this for a long time, very well known in the prostate cancer space. Um, at J&J, all the prostate cancer companies are aware of this target, but it's, again, undruggable. So the investment community tends not to know about it. The physician community tends not to know about it because there's no one working on it.
Yeah.
And so we've now really like leaned into that and have a beautiful program in the drug discovery stage that we're hoping to take forward towards the clinic in the not too distant future. So that's an example of what's coming next. We've shown with that compound some amazing data, and so you'll see that as we start to publish it. There's another prostate cancer program. So prostate cancer is often driven by androgen receptor.
Right.
AR is a principal driver, much like estrogen receptor would be a driver of breast cancer. So despite the products that are currently available for patients, and even those in development that are targeting AR, uh, every time someone becomes resistant to those, the vast majority of these times, it's still using AR. The cancer has found some way to pop out in some interesting fashion to get around the drug and still drive the tumor through AR. So we are working on what I think is a definitive solution with AR, and we'll be able to talk about that in time. But it is something that at Johnson Johnson we dreamed if this were possible, we
Yeah.
bet on AR that would be the thing to do, but that was not possible because it's tackling it in this undruggable site.
Right.
And so we've done exactly that very difficult thing within Parabilis, and that's in drug discovery phase right now, but we'll be bringing it forward. I'm excited about both of these. I think that they're both going to be incredible contributions to prostate cancer care. And then behind that, there are a number of ideas that we're working
Yeah.
on, but again, with the discipline to not expend too much energy on them until we've shown that we've had success with the first few projects. And then you asked a very good question, Pranav, around what would we do with this in terms of licensing? We see so much potential.
Sure.
Are we going to do it all within Parabolis? The answer is clearly no. So we are actively talking to companies right now about use of our platform where we would prosecute the early part of their project, but they would take that project and run with it through clinical development with good economics coming back to our company, but us having an outlet to impact more human disease. And so that's an active conversation right now. We thought of other ways. It's such a good question because I thought about this a lot. There are lots of ways to do that. Could be through licensing the technology. It could be like creating a whole new company and spin out the platform in use in pick an area like autoimmune disease or cardiometabolic disease or neuroscience or whatever it may be. We can create a series of companies or create a series of relationships with other companies that have expertise in certain areas.
You could start a whole venture studio and just find CEOs to run these
We
different
couldn't.
companies.
Yes.
Awesome. And you mentioned a lot about the drug discovery process and the Helicon technology. We have had this massive AI boom in the last few years, and it's hard not to talk about it. Do you feel like the technologies that are coming out with this modern wave of AI have been what enabled this kind of rapid drug discovery with the
Yeah.
Helicon technology and
I
how it?
should have mentioned that AI methodology or computational methods, both physics-based methods, microdynamics type methods, and machine learning methods, classically AI methods, are part and parcel of how the platform works.
Yeah.
So if this whole experimental innovation, set of experimental innovations that we had existed 10 years ago, we couldn't prosecute it. So it's only because we live in this AI era that we've been able to do this. So the way that works is um, I've mentioned that at each spot on this peptide, we can diversify the number of amino acids well beyond the 20 natural. So imagine if you had 1,000 or 10,000 different options for each spot. It's exponential, right? So if you have 20 spots, say, you have 1,000 to the power of 20 or 10 to the 60 different compositions. So there's no way you can experimentally muscle your way through. There's not any chemist in the world that could imagine space that large. So the way we manage it is we made large volumes of training data within the company and use the most modern of AI methodologies to create algorithms based on our proprietary data that are themselves proprietary that then help us with design. So the design features that we're trying to optimize for, first is this gentle entry, this very non-harmful entry that I mentioned, and others can be binding, selectivity, solubility, aggregation, all the features you need in there. So the AI methodologies are part and parcel of Parabilis. Uh, we are a perfectly hybrid company. We are equal parts biology and chemistry, that kind of science, and technology. So it's a modern company in every sense. And I can't imagine doing this at a better time than right now.
You know? I love that. We do wanna be respectful of your time, so we'll wrap up in a couple minutes, but I'm curious, where did the name Parabolas come from?
Ah, very good
'Cause
question.
you said it was renamed, it was originally
It was called Fog Pharma. And so fog, where did fog come from? It was Greg Verdine was the founder, and he was innovative in so many ways. He's absolutely brilliant man. His ideas have seeded a number of companies, including Warp Drive, which is basically the foundational technology of revolution medicine, Wave Pharma, so a number. So, but he wanted to fund this company in an interesting way by friends and family instead of regular venture and called it Friends of Greg, FLP. And so it's an amazing name, and it was carried its day. But fog on a couple levels was mixed up sometimes with Foghorn, which is another company in Boston, and fog sometimes inadvertently conveys a haziness or lack of clarity. So there was a groundswell within the company saying we're turning a page. There's a new chapter. We have clinical, programs now, and so Parabilis, first of all, it sounds like parabolic, and parabolic escape velocity is the velocity you need to escape Earth's gravity. So parabolic implies like shooting for the moon and shooting for the
Mm.
stars. Yet parabilis is a word in Latin that means obtainable, pragmatism. So our company name Parabilis is squeezed between um, aspiration and pragmatism, which I think is perfectly appropriate.
That's beautiful.
Mhm.
That's beautiful.
Very unique name. Just one last question we have for you is, what advice do you have for someone who might be interested in innovating in healthcare, trying to solve challenging problem or develop a new drug themselves?
Yeah, often the temptation is to enter a company and just get a job. But there's so much opportunity right now. Science has evolved in amazing ways. AI is on this massive
Yeah.
upswing at the moment. Uh, I would not do anything but aim high. Look at startups, look at starting your own company. If you go to a larger company, aim to do something really big within that company, use it as a platform to change the world. So I think don't give in to this feeling that you need to just go get paid to do work and aim to change the world.